What plasmids are. Plasmids are small pieces of DNA. So generally, genome can be packed into a big piece of DNA, like a chromosome, which is usually a linear piece of DNA and has millions of base pairs. But it can also be packed in a small circular piece of DNA called a plasmid, and it has only a few thousand of base pairs. And plasmids are of exceptional importance for bacteria. Not really found in humans, but they're of great importance for the survival of bacteria. They are common and widespread. So essentially, a bacterial cell, there's a very simple drawing of a bacterial cell. It has a bacterial chromosome, which has most of the genome of the bacterium, and it has some plasmids there as well, and they can come in and out. They can be exchanged with different bacteria in the environment. They can be swapped between different species as well. They are very important because they can carry virulence factors. They can confer antibiotic resistance. They can code for bacterial toxins, and so on. They're also widely used for genetic transformation because they can be easily engineered. They can put pieces of DNA in and out, and you can make the cells do things with that piece of DNA that you put in. And as I said, the plasmids themselves can be inserted and taken out of cells quite easily. But, importantly, when the cells divide, the plasmids are replicated separately from the chromosome. So so when the bacterium divides, it duplicates its genetic material, and the plasmids need to do it themselves. They can't rely on the cell, the machinery that replicates the chromosome, to do that. And that has a problem. So essentially, we can group plasmids in different categories, but when you look at how many copies of plasmids there are in the cell, there are essentially two types of plasmids, the high copy number plasmids and a low copy number plasmids. And for the division, it poses the following problem. If you have a lot of copies, and then you divide of the plasmid, and then you divide the cell in the middle, and the plasmids just end up stochastically in one of the two daughter cells, it doesn't really matter that much that they don't ensure that they do end up in both of the two daughter cells. However, for low copy number plasmids, that's a bigger problem because if there's just two and the genome, the plasmid is duplicated and then the cell splits, well then half of the progeny of that cell won't have the plasmid. Obviously, plasmid is selfish and it wants to maintain its own genome. It wants to be duplicated. So these plasmids have evolved a mechanism in order to ensure that they are found in both daughter cells. And it is quite simple. It just relies on a protein filament that grows and physically separates the two copies of the plasmid. And it pushes it physically to the two opposing ends of the cell. So this is very simple. It just relies on a protein that polymerizes in such a way that forms a filament, much like actin, for example, in human cells. And then the filament grows and it attaches itself to the plasmid copies and it pushes them apart. Okay? So we can use cryo-EM to see that. So this is a cryo-EM tomogram. So essentially you take a lot of cryo-EM pictures of an object at differing angles, right? Much like a PET scan. In fact, the principle is similar. And then you combine all of these images together and you reconstruct a three-dimensional electron microscope map of the object. And this video here, it just goes, it just goes through the cell and you can see the filament that I was talking about running across from one end of the cell to the other. And that's an E. coli cell which has that plasmid segregation system which I was talking about. It's slightly more complicated than that, but not much. You have the filamentous protein, which is some kind of small protein that is composed, that makes the filament. And you have an adapter protein, which is a DNA binding protein, which also binds to the filament. It tethers the plasmid to the end of the filament. And usually you have two of these coming together, and the filament in the middle grows, and it pushes the plasmids apart. So there are a number of these systems. I just listed here three, And two of them are found on very important plasmids that have been found to confirm multi-drug resistance in both E. coli and S. aureus. So they are of vital importance for medical research. And the plasmid, as I said, the plasmid contains a centromere, which is a DNA, which is a sequence to which the adapter protein binds, and then the adapter protein allows for the growth of the filament. We can see that work in, well, this is a bit dark, but this is a kind of fluorescence microscopy, which is called total internal reflection fluorescence microscopy. And you can see that on the film on the left, which I'm going to play again, you have the filament, which is in violet, and it's pushing two pieces of DNA apart, which are green. And we can also see that these, what we call spindles, so they're like segregation spindles inside the mitotic spindles inside eukaryotic cells, that these are composed of two separate filaments. And if the filaments dissociate, the whole thing disassembles. Okay, so essentially it works like this. You have the binding of the adapter to the plasmid, you seed the filament, so it starts growing from the adapter. The filaments use energy of hydrolysis of ATP for growth. However, they can't continue growing indefinitely. They are unstable if they are unpaired with another filament. So if a plasmid then starts growing its own filament, and if it finds another plasmid that also grows its filament, the two pair together, and that allows for the stabilization of the filaments. The two filaments then slide to minimize the energy, and at this point, so they come closer together, and at this point, they are stabilized, and new monomers can be added from one of the ends, and the overall effect is that the whole thing grows in opposite directions to both ends of the cell. So it's a very simple system. So as I said, we can look at these with cryo-EM. So these are the bundle. So these are we purified these filaments from this particular case, Bacillus satilis subspecies natto, which is a subspecies of Bacillus satilis used in fermentation of soy and making a Japanese speciality called natto. And the plasmid called PLS32, and it contains these filaments that form bundles. And we have shown through cryo-EM that these are in antiparallel orientation. So it means that the protein monomers inside are arranged in such a way that both of the two filaments, top and bottom, run in opposite directions. And that is important for this filament growth that I was talking about, that ensures that both copies are pushed in opposite directions. If it was parallel, that wouldn't work because both filaments would grow in one direction. Okay, so we can also take singles of these filaments, and we can look at the molecular structure, the atomic detail of that. So we can purify them as single filaments, so there are bundles here, but you can purify them as single filaments, and you can then, as I'm sure Wojtek has explained, you can take little pieces of this image from an EM microscope, and you can average them together. And you can from that you can reconstruct the atomic model of the object. So these filaments, they are composed of just one protein that is just repeating in a helix. In this case, this is a double helix. Maybe you can see that. And it's just a repeating object. So taking little bits and pieces of that will allow you to get all of the orientations of that object in the eyes, because it is rotating like a helix. Okay, and that allows us to reconstruct an atomic model. So these are atoms and, inside that object, and I haven't really shown it very well, but the point is that there is a protein monomer, and it's repeated in this helix, and we can show that when you take two of these together, and you arrange them in antiparallel fashion, you can even pinpoint the residues which are important for so amino acid residues which are important for making them bundle. So that's this native property of the filament that allows it to make these spindles that go in different directions. So you can see that. And just the last thing I wanted to say is that there are a lot of these filaments, and they're in all different organisms, and What is interesting about them is they're very evolutionary widespread, so from bacteria to humans. Well, in this case, on the left, you have an actin from the muscle of rabbit, from rabbit's muscle. And then there are a number of other of these filaments, and they all share the same architectural property. They make these long assemblies of monomers, and they twist in different directions. They have different, they're twisted to a different degree as well, as you can see, but the principle is the same. There are some filaments as well that don't twist, like that one on the right. But an interesting thing is that all these proteins are quite similar in structure, but their sequence identity is very, very low. So I just wanted to summarize what I was saying about this plasmid segregation. So what's interesting about it is that it's a very simple molecular machine. So you have just two proteins and some ATP, and it will push two pieces of DNA apart. So maybe you could move something else. Who knows? It depends on how you put it onto something else. And it's interesting that there's some shared architectural principles amongst filaments that have interesting evolutionary points, but I don't have the time to go through that today. That's that. Thank you very much for your attention.